Oh, okay, back for the second half of back for the exciting conclusion of bilateria. So let's cut over to the slides where we were. And let's talk a bit about development. In most animals, and actually you get versions of this even in sponges and cnidarians, Mr. Sperm meets Mrs. Egg, and they get together and make a very special hug. And fertilized egg divides into 2 cells, 4 cells, 8 cells, divides into a ball of cells, and the ball hollows itself out. And a hollow ball of cells is called a blastula. And the blastula then folds into itself to produce an internal pouch that becomes the gut. There's actually several different ways in which this can happen. There's a lot of variations on the theme. But they end with the production of an embryo with this pouch that will become the gut. The process is called gastrulation. And at that stage, the embryo is called a gastrula. So the blastula is basically a hollow ball of cells that folds into itself, undergoes this gastrulation process, and at the end of it, you've got a gastrula. Gaster, of course, is Greek for stomach, so a gastrula is a little stomach thing. And here's an example of what it looks like in starfish embryos. You've got early gastrulation visible on the left. That's a hollow ball of cells. And you can see a dent is forming down at the bottom of it. Looks a little bit like if you took a balloon that wasn't inflated too much and you started pushing your finger into it. You'd make a dent in the balloon. Well, on the left, there's an early stage of that dent. There's a later stage visible on the right. You can see that dent has gotten a lot longer and it's expanding a little bit at the tip and it's forming a definite tube. That sets up 2 of the 3 basic cell layers that bilaterians are made of. The cells on the outside form a layer called the ectoderm, and the cells that form that gut are the endoderm. And you might remember in bilaterians, there's a 3rd layer in between the 2, the mesoderm. The cells that migrate between the ectoderm and the endoderm are what give rise to the mesoderm. And in sea urchin embryos, very close to starfish, the early mesoderm is a kind of loose tissue, cells that are not tightly packed together, that happens to be called mesenchyme. So there you can see there's a blastula, that hollow ball of cells on the left, and on the right, you can see there's that dent forming. The endoderm is forming the embryonic gut, and then those cells that are kind of wandering around on the loose become the mesoderm. Okay, you can kind of see it here if you look over on the left. It may not be super obvious, but I can see some kind of irregularly arranged cells on the left-hand picture to the immediate right of that dent. That's going to be some of that mesenchyme. The starfish and the sea urchin have very similar patterns here. Right. In some bilaterians, the dent is called the blastopore, the place on the blastula where the cells start migrating inwards. So, yeah, I guess I didn't use that term in a previous slide, but the spot on the outside where the cells are wandering in, that's the blastopore. And in some bilaterians, the blastopore becomes the mouth, and the anus has to form later. Often that tube will grow and grow, and eventually it will kind of break through on the other side, and you'll have a tube within a tube, as it were. You have a mouth and an anus, and the anus forms secondarily. Animals that do that are called protostomes. Protos is Greek for first, stoma is mouth. Protostomes means first mouth. This can vary a bit, even within phyla. There are protostomes where the blastopore will actually kind of pinch shut in the center, and so the blastopore becomes both the mouth and the anus. There's some cases of unusual phyla that can vary this a bit. We're kind of generalizing here, maybe a little bit too much, but that's kind of the nice, neat plan. In others, the blastopore becomes the anus and the mouth forms later, and those are called deuterostomes, which means second mouth. Deuteros is Greek for second. Both of these groups are very well supported monophyletic taxa. There's a lot of DNA evidence and some other anatomical and structural evidence supporting the hypothesis that all deuterostomes come from a unique common ancestor, and all protostomes come from a unique common ancestor. Since starfish are deuterostomes, that means that dent down there on the lower side of both pictures is going to become the anus, and the mouth will break through later on. The thing will develop a mouth kind of actually on the side, and the mouth will form second. And by the way, people use starfish and also sea urchin, closely related, use these species a lot in developmental biology because it's fairly easy to harvest lots of eggs and sperm. And if you do it right, it's possible to raise the embryos through a long range of stages. You can see everything from fertilization all the way to, adult sand dollar or sea urchin or starfish. And it's fairly easy to raise, and you can watch the whole thing. The embryos being translucent, you can see things, on them. You can see this process in great detail, and people have been doing that for well over 100 years now. Right. It's not universal, but usually protostomes show a type of cell division in those very earliest stages that's called spiral cleavage. That's actually shown on the bottom. You have a cell, and it divides into 2 cells. And then when it divides into 4 cells, the division plane is at right angles to the first division plane. So you have 4 cells right there that make up a tetrad, a group of 4. And then the third cell division is at right angles to both of them. So you end up with an embryo that has, 4, 2 quartets, 4 cells sitting on top of 4 cells. You can think of it that way. In spiral cleavage, the top quartet rotates about 45 degrees relative to the bottom quartet. So you'll have 4 cells on the bottom and 4 cells on the top. And the cells on the top are kind of nestled into the dents between the cells on the bottom. So that grouping of 4 cells is turned 45 degrees. That's spiral cleavage, and it's kind of shown there in the middle down on the bottom. Deuterostomes will typically show radial cleavage, where the 2 quartets are not twisted with respect to each other. They're sitting directly on top of each other. You'll see this again when we talk about, there's a major group of protostomes called the spiralia that shows this really well. and we'll come back to this. Many protostomes used to be said to have something called mosaic development. Mosaic development means that cell fate is determined very, very early. From the very first cell division, cells are pre-programmed to develop into certain structures. Most deuterostomes were said to have what's called regulative development, in which cell fates are not determined immediately. If you look here on the left, that's a typical larval snail. It's got a foot. It's got a complete gut. It's even developing the earliest stages of its shell. That's a baby marine snail on the left. The thing on the right is a larval snail that's missing a lot of that. You can see it's got some of its gut, but you can see it doesn't have a larval shell. It doesn't have a foot. It doesn't have a lot of things. And here's why. At the top left, you've got a freshly fertilized snail egg. And the first thing that happens is that snail egg develops a bud. This blob of cytoplasm buds off, but never quite splits off, and it's called the polar lobe. And then when the main body of the cell really does divide, it divides into two cells, one called AB and one called CD. And the polar lobe is stuck to cell CD. Whatever's in the cytoplasm of the polar lobe doesn't move into cell AB. It stays part of cell CD. It gets reabsorbed into cell CD, and then it comes out again, and the two cells divide again, and the polar lobe goes with cell D, because you can map these earliest stages of development and tell individual cells apart. And the polar lobe ends up absorbed only into the D blastomere. Whatever was inside the polar lobe does not end up in A, B, or C. The reason that matters is that when this pattern was discovered, naturally people decided, hey, what happens if I do this? And you can actually cut off the polar lobe. It's not that easy to do surgery on embryos, but some species are large enough that it's not too difficult to do. Embryologists used to use things like little loops made out of baby's hair to divide or lop off individual cells from an embryo. And when the polar lobe is cut off, you get the embryo that we see on the right. An embryo without the polar lobe is never going to develop, actually, most of its mesoderm. It's not going to develop muscles. It won't develop eyes. It won't develop a proper shell. It won't develop a heart. All of those things come from that mesoderm layer. And if you remove the polar lobe, the mesoderm doesn't develop, and none of the structures that develop from mesoderm ever develop. So the idea is that the polar lobe contains something. We call them cytoplasmic determinants. They're proteins. I believe they are DNA binding proteins that control gene regulation. But even before that was known, it was known that there was something in the polar lobe that programmed the cells that had it as early as the 4-cell stage. As early as the 4-cell stage, you could tell that cell D with the polar lobe was going to develop into the mesoderm. If you took an embryo at the 2-cell stage and you split it, the CD plus the polar lobe cell would develop into a normal embryo, and the AB cell without the polar lobe would develop into an abnormal embryo. So that's what we mean by mosaic development. the cells are like tiles in a mosaic in that you can't remove them without damaging the total pattern. And the cells are programmed very early to have the fates that they have. Humans, of course, if you take a human 2-cell embryo and you divide it in two, what you get is identical twins. At the 2-cell stage, both of the cells of an embryo are still able to develop into a complete human being. When they do in vitro fertilization, they'll fertilize eggs in a test tube or a dish, I guess, and let them develop to the 16-cell stage, and then remove one of the 16 cells for genetic testing to make sure that the embryo is, genetically normal. And that does not cause test tube babies to be born with only 15/16 of their organs. A 16-cell human embryo can lose one cell and still develop normally and compensate for the fact that it's missing. So humans have much more regulative development because the fates of what our cells are going to become are not fixed nearly that early in development. So yeah, there we've got some actual embryos. You can see there's a 2-cell embryo with the polar lobe, and then the polar lobe gets reabsorbed into that CD blastomere cell. And the polar lobe contains those substances that determine the fate of the cell that it's attached to. Remove those substances and the cell will never develop those set of structures. It's like you've just removed the program. You've just removed the program code. Regulative development is not pre-programmed like that. I already talked about human development, but sea urchins, for example, are also more regulative. In 1895, Hans Drisch was able to take 4-cell sea urchin embryos and separate them into 4 cells. Bit technically tricky, but you can do it. And it turned out that each one of the 4 cells on its own could develop into a more or less normal sea urchin larva. A normal sea urchin fertilized egg will develop into a type of swimming larva called a pluteus, which you see at the lower left. And if you cut apart a 4-cell embryo of a sea urchin, each one of those 4 cells will develop into a more or less normal pluteus. You can't do that with snails because the cell, 3 of the 4 cells don't have those polar lobe cytoplasmic determinants, and any embryo that comes from them is never going to develop any mesoderm. So that's the difference between mosaic development in the snails and more regulative development in sea urchins. We now know, however, that the distinction is not nearly that clear-cut. It turns out that embryos of both protostomes and deuterostomes show both mosaic processes and regulative processes. These are embryos of a deuterostome, a type called a sea squirt, aka a tunicate in the genus Baltinia. There's a lot of work on embryology that's been done on tunicates because tunicates have the interesting property that cytoplasmic determinants are sometimes colored. So on the left, that's a freshly fertilized Baltinia egg, and the cytoplasm, that halo of cells around it, is not part of the embryo, the clear stuff. The egg itself is mostly orange, and you can see at the lower left of the cell, there's a zone of much darker red-orange cytoplasm. When that divides into the 2-cell stage, you can see that that zone of darker orange cytoplasm has been divided into 2. So if you look at the embryo in the middle, Each of the two daughter cells has some of that orange cytoplasm. And then if you look at what looks like an 8-cell stage over there, you can see that some of the cells have that orange cytoplasm and some don't. That orange stuff in the cytoplasm is a cytoplasmic determinant, and it's responsible for muscle tissue. Cells that don't have it will never develop muscle tissue. You could actually take a micropipette and slurp out that dark orange cytoplasm, and embryos that have been treated that way never develop muscle tissue. Because that orange pigment is now known to be a protein, and because it's responsible for developing muscle, somebody named the protein Macho-1. So there, cue the Village People song, if anybody remembers that. You know, Macho, Macho Man, there we go. Or Macho, Macho, tunicate. It's all in that orange-colored protein. And I show you this because that's a mosaic process, because cell fate is determined very early by these cytoplasmic determinants, and yet it's present in a deuterostome. So we now know that both protostomes and deuterostomes can show both mosaic processes and regulative processes during development. The distinction is not clear-cut anymore. Right. Getting somebody to actually teach this in our department has been harder than Hogwarts finding somebody to teach defense against the dark arts. I wish we'd had better luck in finding somebody who could teach it. I mean, I would give it a shot, but I wouldn't be good at it. But developmental biology is a hot field right now, because over the past 20, 25 years or so, it has become possible to start asking and answering very deep questions on the real nuts and bolts of how do you go from a fertilized egg to an adult. You know, there were some really nifty results coming out even when I was an undergrad. Man, I was a sophomore, and we learned about a woman named Christiane Nüsslein-Volhaut, who worked on fruit fly embryology, and who incidentally won the Nobel Prize maybe 10 years ago or so. But yeah, this has expanded a great deal. It's a really hot field, and I wish I knew more about it and could teach it more effectively. Without going into too much detail, what we can say is that development is governed by a network of regulatory genes. And the genes are arranged in a hierarchy. This is a piece of the hierarchy for the development of a fruit fly. If you look at the top level where it says maternal gene products, when mommy fruit fly makes her eggs, even before they're fertilized, she pumps messenger RNA into the cytoplasm of those eggs, and that mRNA is transcribed and translated into proteins. So up at the top, you have 3 proteins, one CAD, that's caudal, one is abbreviated BCD, that's called bicoid, one is abbreviated NOS, and I forget what that stands for. What those proteins do, caudal, bicoid, and NOS, is that they bind to other genes. They bind to the promoter sequences of other genes, and they might either promote them, which is shown by an arrow, or they might inhibit them, which is shown by a line with a blunt, kind of T-shaped end. So bicoid promotes a gene called HB. I think that stands for hunchback. NOS represses the gene hunchback, HG. HB represses and is repressed by a gene on that level called KR. I think that stands for Krupel, which in turn is repressed by a gene called KNIRPS, abbreviated KNI. And I'm not going to be able to talk you through the whole thing, but those maternal genes influence, they promote and or repress a set of genes called the gap genes that determine basically what are the major regions of the embryo going to be. The gap genes repress the Old Navy genes, and Old Navy represses target. No, sorry. The gap genes promote or repress, they can promote and repress each other. And they also promote and repress genes on a lower level of the hierarchy called primary pair rule genes, like even, skipped, runt, and hairy. And those promote or repress each other, and they promote or repress sets of genes called secondary pair rule genes. And those promote and repress segment polarity genes. So the idea is you have sets of genes, each one of which acts among itself and acts on genes at a lower level. So that's what that hierarchy is. And mutations in things like the bicoid protein up at the level of maternal gene products, an egg with faulty bicoid protein will actually develop tails at either end. Mutations in genes that are lower in the hierarchy can result in too many segments or not enough segments or segments forming backwards or something like that. What these genes do collectively is lay down the very basic architecture: which end is the head, which end is the butt, which side is dorsal, the back, which side is ventral, the belly, how many segments are there going to be, where are the segments going to go, and so on. Up there at the top, that's a fruit fly embryo at a very early stage. That may just be an egg that's stained for the bicoid protein. And that bicoid protein is only found in the head end because mommy pumps it only into the head end. That maternal effect gene regulates the expression of gap genes. Down there in the middle, that's an embryo that's been stained for the protein products of the hunchback gap gene in orange and the Krupel gap gene in green. And you can see a yellow zone, which is where they're both being expressed. And then the gap genes regulate so-called pair rule genes that start laying down segmentation, like the gene Fushitarazu, which has been stained down there. So hopefully you can get some sense of how these specify some very basic architecture of the embryo. And both of these gap and pair rule genes regulate yet another set of genes called the homeotic genes. There was supposed to be some slides in there about what homeotic genes do. Let's just say that all animals have some number of homeotic genes, but in bilateria, you have clusters of different types of homeotic genes that further specify the basic architecture of the body. In fruit flies, if you have a mutation in homeotic genes, you can end up with the wrong type of appendages. You can end up with antennas growing out of the mouth, or legs growing out of the mouth, or balancing organs turning into wings or things like that. In you, homeotic genes are involved a lot in regulating how your nervous system develops, which block of your nervous system is going to become your medulla, which block is going to be the cerebellum, which block is going to be such and such part of the spinal cord, and so on. And all of that is regulated by this top-down hierarchy of genes. And I wish I had more time to talk about that. It's a pretty amazing field, and maybe someday we can hire somebody who's qualified to teach that stuff, because it's a hot field, and we're learning a lot of really awesome, very basic, fundamental things about how you get complex patterns in organisms. And this, of course, has some benefits for understanding things like human birth defects. Anyway, okay. Protostomia consists of four main subdivisions that we're going to be going through in agonizing detail. Lophophorata are protostomes that feed with a looping ring of ciliated tentacles that's called a lophophore. Platyzoa are generally small, often but not always there's a jaw apparatus, and usually there's not a functional coelom. The best known platyzoa are the platyhelminthes, that's the flatworms. You might have seen planaria worms before. They're in the platyzoa. Trochozoa develop from a planktonic larval type called a trochophore. We'll talk about trochophores later. And the two big trochozoan phyla are annelids and mollusks, segmented worms, and then mollusks are clams and snails and squid and such. And then finally, ecdysozoa molt a cuticle made out of chitin under hormonal control. That includes the cicadas that are going to be screaming any day now, right? You ever do that? You ever pick up a cicada exoskeleton and like stick it on your finger and run around freaking your little sister out? Not that I ever did that. But yeah, you know that arthropods, like insects, molt their cuticle, and this is done under the control of steroid hormones. All arthropods do this, not just insects, but crustaceans and spiders and such. Roundworms do this, and some other phyla, including the coolest phylum of all, the tardigrada, my personal favorite. Deuterostomia, there's only three major phyla in the deuterostomes. Echinodermata, Greek for spiny skins. The most famous ones are starfish, sand dollars, sea urchins, sea lilies, brittle stars, sea cucumbers. Very diverse phylum in marine environments, although there aren't any in freshwater. The phylum Hemichordata is acorn worms and a very obscure group called the terebranchs. We'll talk about those later. And then the phylum Chordata includes the subphylum Urochordata, which is the sea squirts or tunicates. The phylum Cephalochordata, which we're going to sing about, because every time I teach this, we have to sing the Amphioxus song, and we will definitely do that here. Just because we're dispersed is no excuse not to sing the Amphioxus song. And then the subphylum Craniata, which includes hagfish, and then vertebrata, which is all other vertebrates, for which you can take Biology 4402 if you're just dying to find out more. For that matter, we've got vertebrate zoology, comparative vertebrate anatomy, and then ichthyology, herpetology, ornithology, and mammalogy. So that's what? We've got 6 courses on vertebrates, and we've got invertebrate zoology for the other 97% of the bleeping animal kingdom. Not really fair, but there you have it. Anyway, that's where we fit in. And when we pick this up, we'll talk about deuterostomia first. And we'll start by talking about echinoderms. So that would appear to be that, and that's where I'm going to end it for the day. So I'm going to stop recording. If I can remember what to click, and I'll stop recording now.